Hello everyone. Today we will present a research titled Decoding SARS-CoV-2 Transmission and Evolution. This study looked at the changes in the SARS-CoV-2 virus and how those changes affect the way we test, treat, and protein against COVID-19. This research is about the SARS-CoV-2 virus which causes COVID-19. Our research looked at how the virus changes or mutate and how it spread in different countries. These changes are very important because they can affect how we make tests, vaccine, and medicine to fight the virus. If we understand how the virus changes, we can make better tools to control the pandemic. SARS-CoV-2 is an RNA virus. It is part of a group called beta coronavirus. The virus has a genome size of about 29.99 kilobases. This genome is like a set of instructions that tell the virus how to work. It makes 4 important proteins. First, the spike protein helps the virus enter human cell. Second, the envelope protein helps the virus grow and move. Third, the membrane protein gives shape to the virus. And lastly, the nucleocapsid protein protects the virus RNA and help it multiply. The first complete genome of SARS-CoV-2 was shared on January 5, 2020. This is called the reference genome and scientists use it to compare with other virus samples. You can find it online in the GenBank database. The accession number is NC045512.2 and the website link is shown on the slide. Lastly, closest related organism. SARS-CoV-2 is very similar to a virus called SARS-CoV. That virus caused the SARS outbreak in 2003. Both viruses belong to the same family and have many things in common.
Next, I'll present an overview of the bioinformatic tools we use to analyze the SARS-CoV-2 genome. Next, I'll present an overview of the bioinformatic tools we use to analyze the SARS-CoV-2 genome. Next, I'll present an overview of the bioinformatic tools we use to analyze the SARS-CoV-2.
To begin, we use Cluster Omega, a bioinformatic tool that aligns multiple genome sequences of SARS-CoV-2. This helps us identify mutations and genetics variation between variant strains. It's highly accurate and efficient, especially for a large dataset. However, it has limited customization options and can be memory intensive. This tool lays the foundation for comparing how the virus evolves across populations. As shown here, each viral protein has accumulated multiple mutations, especially in the spike and nucleocapsid proteins. Next, we measure genetic differences between strains using the Jaccard distance. This method looks at the presence or absence of mutation to calculate how different two genomes are. It's simple and ideal for large binary datasets, although it doesn't account for the order of mutation, which can be limiting in some cases. Once we have genetic distances, we apply K-mean clustering, a machine learning method. It groups SARS-CoV-2 variants based on genetic similarity. This is useful to track new variants, especially those that may spread faster or escape immunity. While powerful, it requires us to choose the number of clusters and is sensitive to how we start. Still, it helps uncover hidden patterns in the data. Finally, we use Principal Component Analysis or PCA to simplify complex genomic data. It reduces thousands of variables into a few dimensions so we can visualize patterns more clearly. This helps researchers identify trends and groupings at a glance. Though some data may be lost, it's a powerful tool for insights and communication.
Next, we'll go through in-depth into the methodology used in this research. As we can see, this is the analytical methods used for this research, starting from the genome collection until lastly clustering. We'll go through each of these methods together.
We will move to the data collection and sequence alignment. The first step was to collect the SARS-CoV-2 genome sequences. A total of 15,140 complete and high coverage genome sequences were retrieved from the GISAID database, or known as the Global Initiative on Sharing All Influenza Data. Importantly, a reference genome, specifically NC045512.2 from GenBank, released by Zhang's group at Fudan University, was selected to serve as the baseline for comparing mutations. Once the genome data were collected, the sequences were subjected to multiple sequence alignment using the Cluster Omega tool. The purpose of this step was to ensure that the genomic sequences were aligned in a way that nucleotide position across different strains corresponded correctly to the reference genome. This positional consistency is essential for accurately identifying genetic variations such as single nucleotide polymorphism or SNPs. Then, the next step involved SNP genotyping. Here, the aligned genomes were examined to detect positions where each sample differed from the reference sequence. These differences, known as the single nucleotide polymorphism, were recorded for each genome, including the position of the mutation and the specific nucleotide change. The outcome of this step was a profile for each SARS-CoV-2 genome, essentially a fingerprint of its genetic mutations. The figure on the right indicates the result on the frequencies of the single SNP mutations of SARS-CoV-2 on the genome samples in the world with respect to the reference genome. We will move to the distance calculation and dimensionality reduction. The genetic similarity or dissimilarity between each pair of genomes was calculated using the jagged distance. This metric compares the number of shared SNPs to the total number of unique SNPs between two genomes. Specifically, the jagged similarity is computed as the ratio of intersecting SNPs to the union of SNPs, and the distance is 1 minus the similarity value. A lower jagged distance indicates a closer genetic relationship in evolutionary. Due to the high dimensional nature of the SNP data, dimensionality reduction was performed using principal component analysis or PCA. This statistical method compressed the data into fewer dimensions while retaining the most significant genetic variants. By projecting each genome into a lower dimensional space, we make the dataset computationally manageable and visually interpretable. Lastly, the K-means clustering algorithm were used to classify the SARS-CoV-2 genomes into genetically similar groups or subtypes. In this analysis, two types of representations were used, one based on jagged distance and the other on the location of mutations. To determine the optimal number of clusters, the ALGO method was used. This method involves plotting the within cluster sum of squares against the number of clusters and identifying the point at which adding more clusters no longer significantly reduces variability. The outcome of this step was the formation of genetic subtypes of SARS-CoV-2, offering insight into its transmission pattern and evolution over time.